We're understanding a lot more, and some of it's coming from the genetics as we figure out the genes when it runs in the family that are involved, and then we're studying the genes and trying to understand how and what they do so that we can learn more about the biology of the disease. For instance, you can get frontal temporal dementia from a mutation in the progranulin gene. And progranulin is sort of a growth factor, actually, which is really interesting because if you have a mutation that upregulates it, so you get too much of it, people get certain types of cancer. They're at risk for cancer. And if you have mutations that downregulate it, you get frontal temporal dementia. You can get forms of dementia. So, it's almost like if we can figure out more about it, we can, downregulate it in the high risk for cancer groups, upregulate in the high risk for FTD. We would go for cortical basal as well. So there's a lot, we've really gained a lot in our understanding of what goes wrong in the diseases. So I think there's a lot of hope on that front. You said that your mom was misdiagnosed initially, and a lot of people are in fact, most people I see with cortical basal, and actually most with frontal temporal, are misdiagnosed. I'm their second, fifth, or 20th physician that, opinion. And so we're also in need of what we call biomarkers, better ways to make the diagnosis. And there are things in research that aren't, commercially available yet, but maybe in a few years that pick up, they can image tau, the protein that goes awry in a lot of these cases in the brain. And they're doing it for Alzheimer's because that protein goes awry as well. Alzheimer's, they have two proteins, they have amyloid and tau. And in cortical basal, they just have the tau. And of course, they're developing these things for Alzheimer's, but the ones that target an overexpression of tau might be useful for diagnosing cortical basal. So the research that's going on for Alzheimer's may be very relevant to FTD and cortical basal. And not only would it help give you the diagnosis, because the distribution would be different from the Alzheimer's and they wouldn't have the amyloid, but it might also help you to determine biologically which subtype of these conditions, because they're different. There's too much tau, too little tau, ubiquitin. There's all sorts of complicated biochemistry, but your treatments down the road are going to depend on the biological subtype probably. So that's an exciting area.